Hello, good morning to everybody. Welcome to this SEGNA's second webinar of this year 2020 about natural colors. My name is José García Reverter. I am the director of R&D of the SEGNA group. As you already know, probably SEGNA is a business group with 60 years of experience in the production of natural colors, with production plants in Spain, Turkey, and Italy, and customers in the 5 continents. In today's webinar, we will talk about vegetable extracts and foodstuff based on anthocyanins and betalains. This webinar is expected to last approximately 1 hour with an additional 10 minutes for questions. Today, we have 2 special guests from the academia, Dr. Antonio Moratas and Dr. Fernando Gandia. During the webinar and after this short introduction, Professor Moratas will tell us about the anthocyanins, from where they come, what their stability is, and he will explain to some of their applications. Dr. Antonio Moratas is professor of the food science and technology at the Polytechnical University of Madrid since 2004. Dr. Moratas is also professor in several national and international masters and seminars. He's a big expert in polyphenols, and he collaborates with the Spanish and European administration as expert in polyphenols and natural products. Dr. Moratas published more than 70 international papers, and he is author and co-author of several books related with the polyphenols. Next, Professor Fernando Gandia will talk about betalains, another very important and large family of natural colorants of vegetable origin. Dr. Fernando Gandia is Vice-Dean of Biotechnology at the University of Murcia and professor at the Department of the Biochemistry and Molecular Biology. Professor Gandia Herrero has participated in multiple research projects, and he published more than 50 scientific papers. And actually, and nowadays, his current research lines are focused in betalains, in metabolism of the plants, with a special emphasis in this kind of very interesting colorings plants. After that, Dr. Juan Mario Sanz will talk about the industrial application of the different extracts and juices and their quality control. Juan Mario is doctor in food science and technology from the Polytechnical University of Valencia. He worked in different companies, food companies mainly, and public research organizations. He has published several papers in different scientific journals, and he collaborated in the writing of several chapters of food technology books. Juan Mario is working with us from 2015 in the development of new products in the SEGNA Group headquarters in Valencia, Spain. And finally, we will end with question and answer time. For approximately 10 minutes, if it is possible, we will answer the question that you sent us through the chat. You can send the question during all the presentations. I remind you that this webinar is going to be recorded. This means that later the recording will be at your disposal. In addition, in the coming days, you will receive a copy of the documentation if you need to use them in the documentation that we are going to use in the webinar. But before we start with our guest, I would like to share with you some interesting facts and figures about natural colorants and the market evolution. First of all, I would like to underline that the conclusions and data that we are going to see next were published by reference companies at Maintel, Passport, or Future Market Perspective, among others, and that we have only collect them. As you know, color is essential in the food industry due to the great influence of the consumer perception. If the color is right, the product will be attractive, but otherwise, the consumers will reject it. And as you also know, consumers prefer natural products because they believe they are healthier. And for that, during the last years, there has been a growing demand for natural foods. first in Europe and the United States, but now all over the world. And in the case of natural colors, even more due to the rejection of synthetic dyes, probably due to the consequences of the Southampton report. But in any case, it doesn't seem totally clear what a natural coloring is. In fact, there is not universally accepted definition of what a natural colorant is, and each health agency from different states has his own criteria. Perhaps for this reason, NATCO, the Association of Manufacturers of Natural Colors, has established some guidelines to clarify the situation. You have here the link to the website if you are interested to know more about this. Basically, there are 4 different categories for the different naturalness of the different colorants. And more recently, but also related to the interest in natural products, we see how the term of clean label is being increasingly demanded by consumers. And in this sense, to achieve a clean label product, we have, in fact, only few options. We can reduce the number of ingredients, or at least clarify the source of this. In any case, natural colors cannot be used in a clean label product, unless in Europe, because they are considered additives and the E number must be used. It's perhaps for this that food manufacturers are using more and more coloring foodstuff instead of natural colors. Another interesting fact is that, based on the recently published articles, it appears that the current health crisis is increasing even more the interest in natural and healthy products. Pandemic is accelerating the naturalness movement with an increasing demand for natural foods. And if until now the consumer was interested in their health, now they are more even. And credibility and traceability of the food chain is essential. That is why natural and sustainable is on the rise. To conclude, I would like to present some graphs that can illustrate, perhaps, the evolution of the natural colorant market in the last 6 years and what is expected to happen in the next few years. In the graph number 1, in the first graph, you can see in the left, in the top, today you see that the 70% of the foods use some type of coloring, being only 30% in which they don't use any coloring agent. In the graph below on the left, you can see the relative importance of these different colors and the evolution over the time. Note that the most used colors are brown and black, followed by yellow. Red tones are in the 3rd place, while other colors are consumed much less. In the graph 3, the graph at the bottom right shows the origin of the food colors consumed globally. It should be noted that 27% of the dyes are of natural origin, while 41% of the dyes are synthetic yet. However, as can be seen in graph 4, the demand for natural colors has grown steadily in recent years, mainly used in the preparation of carbonated beverages, pet food, and in the preparation of ready-to-drink teas, as you can see in the graph number 5. Finally, in the graph 6 and 7, you can see how the forecast for 2028 is that the carotenoids will be the natural colorants with the highest profitability and growth potential, closely followed by anthocyanins. And the beverage, packaged, and frozen foods will occupy a prominent position in the use of natural colorants. To finish my presentation, as summary, we can say there is no official definition of what a natural color is by national agencies, but the trend of natural is growing in all markets, so the use of natural colorants in food industries becomes essential. Consumers are pushing to eliminate synthetic colors, mainly in the USA and Europe Union, but not only, and expectations in Asia Pacific, Middle East, and Africa are very high. A significant increase in the use of coloring foodstuff is prohibited because they allow clean labels, no E number needed, and finally, carotenes for yellow and anthocyanins for red are the natural colorants with the best prospects. This is all from my side. I hope you found this information, this whole presentation interesting. And if you have any questions, we will answer them at the end of the presentation. Next, I give the floor to Dr. Antonio Morata, who is explaining something more in detail what is the anthocyanins and how they can be used in the food industry. Thanks a lot. Antonio, please.
Thank you so much, Jose. Thank you to everybody for participating in this seminar, and thank you so much to SEGNA for inviting me to speak in this seminar. I am working in anthocyanins during more than 20 years, especially focused in fermented beverage, but also in other applications. I'm going to explain a little bit about origins, stability, and applications of anthocyanins. Okay, anthocyanins are flavonoids that absorb about 400, 600 nanometers, depending on the substitution in the A and B rings, and also depending on the substitution in the glycoside moiety. They produce colors that range from red-orange to blue-violet, so they cover whatever color that we normally use in foodstuffs. The colors is due to the absorbance in the heteroaromatic ring, due to the electrons that are moving on that. Anthocyanins are responsible of the color of most of the flowers, fruits, and vegetable tissues. They have been studied because they have a lot of possibilities as food pigments. Because they are innocuous, they can be used without any problem, and they are safe molecules also with positive repercussions in health, because they have antioxidant properties, but also they have positive applications in several typical illness. In grapes and wines, they are really interesting because the high relevancy of color in this beverage. You can see on the right side how most of the flowers have anthocyanin pigments with colors that range from yellow to pink or red. Also in fruits, from blue, dark blue, like black blue, and also different scales of red colors. Also, you can find orange and many other colors in some carrots. This is the main structure of the anthocyanin. This anthocyanin has a beryllium cation. This ring is really important in the color and also is the responsible of the color, and also is associated to other aromatic ring. This double ring is called benzoberyllium and is responsible of the color. Also, they have other substitution here with other polyphenolic ring, also with two substitutions in R1 and R2 affecting the color of the global molecules. Anthocyanins many times can be glycosylated in the position 3 and in some cases in the position 5. And when the molecules is without the glucose, is called anthocyanidin. With the glucose is anthocyanin. Okay, in many products, in many liquids, anthocyanins experiment a lot of reactions. Most of the color is due to the cationic beryllium anthocyanin, to the flavillum molecule, and this molecule can evolve to blue forms to blue pigments at high pH, forming quinoidal bases, but also can evolve to colorless molecules like the carbinol pseudobase or the chalcones. These colorless anthocyanins are in equilibrium with all the other molecules to keep in solution the suitable amount of flavinium salt that is responsible of the color. On the other side, some additives strongly used in food, like sulfites, can affect the color, producing the bleaching of anthocyanins, and also the anthocyanins can polymerize with other flavonoids, forming polymeric pigments. Concerning the color stability, you can see that the color of anthocyanins depends on pH, hydration, sulfur dioxide, oxidations, and also copigmentations. So studying this reaction, we can keep them more stable, and we can preserve the color during the aging, during the evolution of the foods. You can see that anthocyanins are strongly depends on the pH. Malvidin 3 glucoside is one of the most important anthocyanins in many fruits. And you can see that at pH 4, the color intensity at 520 in the observed red is very low. However, if we decrease the pH at 2.2, the intensity of absorptions increase a lot. So the low pH strongly affect the color intensity of anthocyanins. Also, you can see how several reactions affect to the anthocyanins. All these test tubes has the same amount of malvidin, about 10 milligram per liter, and you can see that in strongly acidic conditions, the color evolves to red by hyperchromic effect. At high pH, it evolves to blue by the formation of quinoidal pseudobases, but also copigmentation processes produce the effect of biochromic sieve, affecting the color, producing red-bluish color, but also hyperchromic effects. Okay, you can see how the same anthocyanin amount can experiment biochromic effects with a higher wavelengths and higher color intensity. It means blue color or red-bluish colors, and this is why copigmentation also decrease the color by hypsochromic effects in some situations, like for example with sulfur dioxide, or it experiment hyperchromic effect at low pH. Other important reactions is the bleaching by sulfur dioxide. Sulfur dioxide is a strong nucleophilic reagent that can produce the addition to carbon-4, destroying the aromaticity and therefore producing colorless molecules. On the other hand, water, also as a nucleophilic reagent, can be introduced in carbon-2, producing the breakage of the aromaticity, and again, these molecules is colorless. So many reactions affect the color of anthocyanins. Also, it's very important the stability concerning the number of resonant forms that can have this heteroaromatic ring. Other interesting process is the intermolecular reactions, in which the anthocyanin is quite close to other molecules that behave as copigment, producing a modification in the maximum of absorption, normally with biochromic effect, higher wavelengths, higher color intensity. You can see how is the normal evolution in pH of malvin from strongly red to pale colors, from low pH to neutral pH, and also the same concentration with a copigment, like for example caffeine. We can use these reactions in energetic beverages, produce higher color intensities at many pH, but also a biochromic effect with red-bluish color that are really interesting for this kind of beverages. Well, concerning the grape and fruit profiles, you can see that the cherry fruit has a very simple anthocyanin profile with a low amount of anthocyanin molecules. However, a complex fruit like grape can produce more than 17 anthocyanin molecules with 5 single anthocyanins, with 5 single anthocyanins like delphinidine, peonidine, petunidine, malvidine, and cyanidine, but also acylated and acetylated forms. Okay, here you can see a fragment of history. This is the first anthocyanin separation by HPLC, published by Wolf and Nagel in the American Journal of Phenology in 1978. You can see the separation of main grape anthocyanins, where the malvidine 3-glucoside is the main peak of the chromatogram. Also in this part, you can see the acylated, the acetylated anthocyanins, and here the cumarylated anthocyanins. The acylated anthocyanins are quite interesting because they experiment more apolar chemical conditions, and also they are more stable in water solutions, but also they have red-bluish colors. Okay, the substitution in the B ring affect the color. You can see that the 3 substitution in delphinidine, petunidine, and malvidine compared with the B substitution in cyanidine and peonidine produce bluish colors, red bluish colors. Normally they absorb delphinidine, petunidine, and malvidine at higher wavelengths. Also, the acylation produces bluisher anthocyanins. So the malvidine alone is 528 red, and acylated with acetic acid is 533, and with picumaric acid is 535. So we are moving from red to red bluish colors just by the acylation in the glucose. Okay, anthocyanins are normally in the external surface of many fruits, also in the pulp, but it's quite usual to be located in the external surface because they are used as an evolutive strategy by the plants to attract some animals to spread the fruits. So in many fruits, the anthocyanins are strongly distributed in the external surface of them. Even when it's in the pulp, like in the strawberry, you can see that the density of anthocyanins is higher in the external surface because this covering of anthocyanins make the fruit attractive for the animals. We can see that is the same strategy we are using with the beverage and with the covering of many foodstuffs to produce more attractive foods. Okay, also something concerning my research, I have been working a lot in enology to keep stable the color of the wine, and there are several parameters that strongly affect the color stability. We can use selected yeast to produce more stable pigments, and also we can use these reactions to produce more stable pigments in other food products. For example, pyranoanthocyanins that can be promoted like vinyl phenoladox with some yeast that have hydroxythinamate decarboxylase activity, but also the production of other pyranoanthocyanins like betalain can be produced by the release of acetaldehyde and pyruvic acid during the fermentation by yeast. Also, some yeast can produce malic acid or lactic acid, especially some non-Saccharomyces yeast, affecting the pH and therefore promoting the formation of hyperchromic effects, so they increase the color intensity of anthocyanins. Also, we can see lead yeast with low absorption of anthocyanins, so it helps to keep more anthocyanins in wines. Other specific biological agents, as lysagin, can keep stable more anthocyanins because it prevents the oxidation of the anthocyanins. Take a look on this stable pigment promoted by yeast, but also can be formed by some natural chemical reactions. In pyranoanthocyanins, we have a double pyrano ring, so the number of resonant forms is double than in other normal pigments. So the oxygen, and the positive charge in the oxygen can be stabilized in these two oxygens. So the number of resonant forms is double that in a normal anthocyanin. Another interesting thing is that the position 4, that is the more possible for the reaction with sulfur dioxide, is completely saturated. So sulfur dioxide, sulfites, are not able to join as in natural grape anthocyanins or natural fruit anthocyanins, because this position is protected from an extra bond with other carbons. So this position is protected for sulfur dioxide bleaching. So these molecules are not affected by sulfur dioxide. It's quite interesting as well, the color of this new pigment, like betacin-A or betacin-B. We can see that betacin-B is a pyranthocyanin with a lower color wavelength, so it means that they are a little bit red-brown. But betacin-A has a similar color to malvidin-3 or glucoside. Malvidin is about 528 and betacin-A is about 515. Okay, also you can find complementary information about the interaction of color by anthocyanins in wines and juices by reading these two documents. Is my chapter in the book Grape and Wine Biotechnology. Is open access and freely accessible by this link, and also this review article that is focused in the use of non-saccharomyces, Saccharomyces and non-saccharomyces yeast to produce stable pigments. Also is available in this link and is open access, freely accessible from internet. Okay, now we are going to comment shortly some applications of the use of anthocyanins. For example, this is some trial we developed together with SEGNA during a master thesis to use anthocyanins, natural anthocyanins coming from grape concentrates that are produced by SEGNA in fermented milks. Typical yogurts produced by lactic fermentation, and you can see how the color is evolving depending on the pigment used, but also to the addition of some other molecules, like, for example, acetaldehyde, pyruvic acid, and caffeine. In the case of caffeine, we are promoting copigmentation reactions. In the case of acetaldehyde, we are promoting the formation of a pyranoanthocyanin pigment, betacin-B pigments, and in the case of pyruvic acid, we are promoting the formation of betacin-A. So in these two cases, we are forming a stable pyranoanthocyanin pigments by the addition of a small amount of acetaldehyde and pyruvic acid. And in this case, we are promoting the formation or the production of bathochromic effects by copigmentation. You can see how the color is strongly different in the soft background that has a yogurt, but is strongly different by the addition of these copigments and additives. And we can keep more stable along the time. So we can use this reaction to produce new pigments and new color in the yogurts. Sorry. Also, we have used natural anthocyanins to produce vermouth beverage by the addition of natural anthocyanins. The normal pigment that is used in the production of vermouth is caramel. Caramel produces a brown color and is less attractive than red wine color. So when we put in vermouth wines, in vermouth beverage, anthocyanins, natural anthocyanins, the red color is much more attractive. You can see in the commercial product, and more attractive to the consumer. Also, the stability is quite good along the time. We have this kind of vermouth produced during 5 years and with suitable stability of anthocyanin during around 5 years. Lastly, also anthocyanins can be used to produce other fermented beverages like beer. It's interesting to add anthocyanins to beer or ciders to produce new aspects, new products and new sensations in the consumer. So the consumer can take new beers with red color, blue color, orange color, and it is possible to make this beverage more attractive or in any case different for the consumer, so they can select new properties on these beverages. And finally, let me conclude just saying that anthocyanins is an open door to new kind of foods, because the color is the first perception of the consumer. So what the people see, if it's attractive, is a really interesting possibility to promote our new food. Thank you so much, and you can contact with me if you want in whatever of the links that are in my last slide. Thank you so much.
Thank you to you, Antonio, for this very interesting presentation. In fact, several attendees have sent to us some questions about your presentation. But I remind everybody that during the question and answer time, we are going to discuss and to have the opportunity to discuss about the question that they sent us. And now it's time for Dr. Fernando Gandia, who is going to speak about betalains, the origin of betalains and their uses. Fernando, it's your time.
Thank you very much. First of all, let me thank Secna and thank Jose for the kind invitation to talk to you today about this interesting group of pigments, which is the betalains. Betalains are experiencing an extraordinary increase in the interest among the scientific community, and I can anticipate that this interest will move to the food industry shortly. Let me introduce my group. I belong to the Department of Biochemistry and Molecular Biology at the University of Murcia, and there we work in the fields of biology, chemistry, but always with the food perspective. We work in plant metabolism with particular emphasis in pigments, in the enzymology, in the reactions that are leading the production of the pigments, and in the characterization of bioactive compounds in general, with special interest in betalains in this moment. We have what we call the bioactive compounds laboratory, where we work with plant extracts and bioactive molecules of natural origin, and we work in the biochemistry of these compounds, studying how they are produced, how they degrade, and changing this information, using this information for biotechnological applications. Furthermore, we work in the evaluation of their biological activities, and we are developing models of anti-cancer activity that we are applying now to the characterization of the betalains and to other compounds. For this, we use a model animal, which is C. elegans, which is a small worm that we use to try all the bioactivities that are claimed for betalains and other compounds in vivo, and moving from in vitro to in vivo assays. Talking on betalains after the brilliant presentation of Antonio on anthocyanins, I can say that they are very similar pigments at the physiological level, and under certain circumstances of pH, mostly, they share the color, and they have a very similar absorbance spectrum, and they share common physiological aspects in their production, in their location, or in their roles even. So we can find them in nature in berries, as you can see here. We can find betalains in flowers, as we can find the anthocyanins. We can find them in fruits, as cactus pears, prickly pears, or we can find them in beetroot, which is the main product used by the food industry as the main source of betalains. But they are present in most plants of the Caryophyllales order, where anthocyanins are not giving color to their structures. The important thing about the betalains is, at the structural level, is the betalamic acid moiety. There's a portion of the structure which is common to all the compounds. Every single betalain has the betalamic acid moiety, which is this structure that you can see here. And depending on the rest of the molecule, we can have different colors. So we can move from yellow betalains, are this one here, which is which we call the beta-xanthines, or they can produce the violet pigments, which are the beta-cyanins. They are different only in this small portion up here, so we can move from a yellow color to a violet color just by adding different structures at the biochemical level. These differences are naturally produced in the plants. So in plants, we can find yellow to violet colors, ranging in the middle with oranges and red shades. At the synthetic level, I can say that our group has participated in the elucidation of this biosynthetic pathway, which is a biosynthetic net, where we have contributed to the elucidation of the reactions leading to the production of beta-xanthines and leading to the production of beta-cyanins, the violet pigments, by studying all these chemical reactions within the plants. The important thing here, one of the most important enzymes here, is this one, which is a DOTA enzyme, which transforms colorless compounds, tyrosine, an amino acid, and DOPA into a yellow color, which is betalamic acid, which is the structural unit of all betalains. It's yellow in color, and it forms the yellow betalains, beta-xanthines, or can go through these other reactions to form the violet pigments, the beta-cyanins. Afterwards, beta-cyanins can be glycosylated, can be decorated by different chemical structures, all within the plant. But the color is already there. So the violet color is here, and the yellow color is here, coming from betalamic acid, coming from DOTA enzymes. So what we did from the biochemical point of view was to characterize this enzyme for the very first time in beets. So we took the enzyme from beets, we put it into a microbial factory so we can produce the protein, we can extract the protein, we can study the protein, and here we see how we make E. coli extracts without enzyme. We order the microbial cultures to produce the plant enzyme. We produce and purify this single protein, the DOTA enzyme, and we see how this enzyme is able to transform the colorless compound DOTA into betalamic acid, which is already yellow, but is the starting point to make all the other pigments, all the beta-xanthines and all the beta-cyanins. And what we see after deep characterization of the reactions is that we can make in vitro exactly the same thing that the plants are doing. So we have the same pigment, we have the same distribution than in our in vitro assays that we can find in nature. So as an example of the biotechnological applications of our research, I bring to you here the quinoa grains. Quinoa grains are in nature are pink, are violet, are red, are orange, and are yellow. So what we did, and all these colors are due to the presence of betalains in the outer layer of the seeds. So what we did was to go from the seeds to the small plants, to the plantlets, and from those plants we can make cell cultures, and we can order at certain moments that these cell cultures produce betalains, produce a change of color. So we experience an increase of color in cells at the cell level, and the yellow cultures could be used to produce yellow betalains, and pink cultures could be used to produce beta-cyanins. Furthermore, we're interested, all of you, I guess, in the stability of these pigments, in their color evolution, in the influence of different media and different parameters on their appearance in their color. And we are also performing different analyses and studies related to the stereochemistry of the pigments. The most important thing here is that we can make different shades, new colors, just by mixing different compounds of the same family. So just by putting together betalains of yellow color and betalains violet in color, we can make all the different shades that are present in nature. And we can select which pigment can be used for doing this task. For example, we can use the most antioxidant pigment, the most reactive compounds detected in vitro in antioxidant assays. These compounds are not very stable. They are very strong antioxidants, but they are not very stable. So if we can put them together and we can encapsulate them in different matrices, as we did with chitosan or maltodextrins, we could increase the lifespan of these of the products containing them up to several months. Now, remember to you that these are not the most stable, but they were selected because of being the most antioxidant ones. Other compounds, less antioxidant but more stable, can be stored even for longer. And here we present all the different shades, all the different colors that we were obtaining with this technology. Back to the biochemical assays, if we have an enzyme that is able to produce color compounds just from colorless chemical compounds, what we did was using all the biochemical information we already had, put it into biotechnological applications. This way, we were able to make cultures that were natural producing the betalains. Here, we were able to direct the production of different pigments so we can obtain different shades, different colors. And then we can scale it up. We can make it in large volumes and to obtain a huge amount of pigments that we later on can use to purify, to concentrate, and to characterize. So our experience is to take all the sequences, take all the capabilities that we can find in plants and to put it into bacterial cultures, and then scaling it up to the level of bioreactors, and then to obtain the pigments we can work with. Why should we be interested in betalains? Okay, currently there's an interest in betalains because of their antioxidant power. They are very strong free radical scavengers. They produce novel colors and novel shades we've seen. In addition, they are fluorescent, and they are very interesting from the point of view of the pharmacological targets that they direct. For example, they are able to inactivate the inflammatory response by modulating the activity of certain enzymes. In addition, they are anti-cancer molecules. This has very strong potential for betalains as a nutraceutical, and in our opinion, it makes a very bright beginning, new beginning for betalains. On the other side, there are difficulties in demonstrating the health claims of betalains due to the use of extracts, plant extracts, mixtures of compounds instead of pure compounds. The demonstration of the health claims to the regulatory agencies is difficult in these conditions. So what we did was to fill this gap by using an animal model, by using a worm. What we did is to obtain all different betalains, pure compounds, and all these compounds, we were using them in an animal model, as I told you before, this small worm. This is a small worm, only 1 millimeter long, that we can use to try all the different betalains, to try all the activities that have been described in vitro. The good thing about the worm is that it's a complete animal. It's very similar at a biochemical and genetic level to all other animals, mammals, including humans, and it has been demonstrated, is already validated as an animal model. In fact, the Nobel Prize was awarded in 2002 to the people who were describing C. elegans as an animal model. All this technology relies in the measure of lifespan as a measure of the health promoting potential of molecules, extracts, and foods. As any other animal, when the worms are healthier, when the worms are happier, they live longer. So if we measure the life of the worms, we can have a measure of the health promoting potential of the different extracts, foods, in this case, the betalains. So what we did was to obtain a new piece of equipment, which is called the lifespan machine, that was developed at the Harvard University, and that we have replicated in the University of Murcia. There's only a few of these machines in the world, and we have one of them. So what this machine makes is to follow every single worm that we have in small plates. Into account that in each plate we can have a single molecule, that we have a single condition, that we have a different concentration for each one, and that we can have many, many replicates. So what we have here is 50 worms in each plate. We have 16 plates in each scanner, and we have up to 10 scanners going at the same time. So we can follow 8,000 worms at the same time with different molecules, known molecules, different extracts, different fruits, different concentrations. So it's like having a huge hospital with patients that we can trace, and we can follow the life of every single worm. the movement of every single one with the special software produced for this application. What we have here is that the betalains are taken by the animal. Animals eat the betalains, and we can see them because the animals are transparent, and the betalains are pigments, and the betalains are fluorescent. So we can follow the fate of the betalains inside the animal. So the digestive tract is crowded, is here filled with the betalains. We can see them. So they are eating them, and after eating them, it promotes certain health-promoting activities. If they are happier, if they are healthier, they live longer. So we can see here how an animal treated with a single, only one betalain, can live longer under certain circumstances. The best one here in this experiment, it was a small experiment we performed, was in the casanthin, which is the main pigment found in cactus pears. It's a yellow pigment. So we can see how the betalains present in cactus pears make a difference in terms of lifespan of the worms, in terms of health-promoting potential. If this pigment is present in yellow cactus pears, we can take the cactus pears, we can make extracts, and we can see if the extract, the food, is doing the same into the lifespan of the worm. And it did. So at certain concentration, very low concentrations of pigment of the food was enough to see the same increase. Take into account that we are going from 9 days to up to 11 days of life. It's a huge increase. 20, just imagine 10, 15, 20% increase in lifespan of an animal, including taking the reference, for example, of the humans. Okay, so we have the capabilities to obtain every single betalain because of our studies in the biochemistry of the betalains, of the biosynthetic pathway. With this information, we can make every single pigment. Okay, so what we did was to obtain every single pigment present in nature and to study the effect of every betalain in the world. And this is what we had. We had an increase of up to 15% for certain molecules. So they live longer, they're happier, remember, and they have characterized it in this way in vivo, a health-promoting effect of the betalain. But not only lifespan can be measured. We can see the ability to reduce the oxidative stress of the betalains or any other molecule. Betalains, when are administered to the worm, are eaten, are incorporated into the body, and they reduce the oxidative stress. And this can be seen just by tagging those proteins that make the response of the animal in vivo to a fluorescent tag. So we can see, this is the word, we can see how the stress, the oxidative stress is reduced. And here you have the graphs, how betanin, the main pigment present in beetroots, but also in the casanthin, the main pigment present in yellow cactus pears, reduce significantly the oxidative stress. And this is done with a lot of animals, so this is statistically significant. Remember that we had up to 8,000 animals going in the experiment at the same time. Not only oxidative stress, but also anti-cancer activity. If we perform the same kind of assays with a lot of different molecules, different betalains, we can check which betalain is most active against cancer. We can make worms to have tumors, and those tumors can be reverted, can be decreased by the ingestion of betalains. So betalains reduce in vivo the size of tumors in animal models. And the most active betalains have been patented. The use of those betalains have been patented by our group very recently, but not only betalains, purified betalains can do the job, but also food extracts or different plant extracts. All this is linked by the nutragenomics information that we are getting. All these activities displayed by betalains in vivo can be traced up to the level of transcription factors inside the body. When the betalains are injected by the worms, they produce modulation of DNA, they produce the modulation of biosynthetic pathway, they produce the modulation of different reactions. And this can be seen by DNA biochips. So we can trace and we can put together all this information, and it's justified the increase in the lifespan, and it's justified the reduction of the size of the tumors in the animal model. Not only betalains, but also other compounds have been tested with this animal model, flavonoids, polyphenols, other molecules, including drugs that are currently used in therapies against cancer have been tested, and it's working. I mean, an animal model which is being treated with a drug that is used in people is also being reduced, is also reducing the size of the tumors when they are eating these food products. They are eating extract, they are eating foods, and they are eating the betalains, the molecules. All this information can also be translated to other models, animals, models of Alzheimer or models of Parkinson, and all related with the ingestion of different extracts and different foods. All this information of this bioactive potential of betalains making a huge impact in the scientific community. There is increasing interest in the scientific community on the bioactivity and the bioactive potential of betalains, and we consider that this interest will move into the food industry because of the health promoting potential of the betalains. As a summary, betalains biochemistry give us the clues on how they are produced in the plants. We can produce them using this information at a biotechnological level. Multiple shades are possible if we mix together different types of betalains. The stability of the betalains can be increased by studying the parameters by encapsulating them. And the model animal C. elegans makes us, for the first time, to see how each individual betalain or how each extract or how each product is performing a very huge impact in the lifespan and on the health promoting potential of the betalains, moving now from science to the food industry. I would like to thank my group, the Biochemistry and Site Biotechnology group at the University of Murcia, for all the support, all their work, and to the funding bodies that are behind our investigations. And thanks to you for your attention. Of course, I'm at your disposal for any extra information or inquiry you might be interested in. Thank you very much.
Thank you to you, Fernando, for this very, very interesting presentation. I will be listening for a long time your presentation. And if you want, of course, we will have the opportunity to have more in the future webinars. And I would like to have your presence in these future seminars.
Of course, no problem. You're welcome.
Now, I give the floor to Juan Mario Sanz, who is going to talk about the industrial application of the colorant extracts, and he is going to discuss about the different strategies and alternative for the quality control, the most important quality control parameters of these products. I give you the floor, Juan Mario.
Hello, good morning. First of all, I want to thank you very much to Professors Antonio Morata and Fernando Gandia for these very interesting presentations. Today, I'm going to talk about the industrial applications of coloring extracts and juices, and which are the most important parameters regarding the quality control of this type of products. Here, I show you a brief overview about the contents of the presentation. First of all, we will talk about the anthocyanin extracts. Then we will continue with fruit and vegetable juices that have high amounts of both anthocyanins and betalains. And finally, we will see some applications in the food industry. Anthocyanin extracts are natural red colorants extracted and purified from many fruits and vegetables. Due to the high color intensity, these pigments can be successfully used to color food and beverages. When they are used as food coloring, have to be identified with the in number 163, according to European regulation about the specifications for food additives. Here at Segna Group, we are dedicated to extraction and purification of these colorants by an exclusive technology that gives us a very high quality to our products. We are specialized in the extraction of anthocyanins from 3 natural ingredients as a raw material, such as grape, black carrot, and red cabbage. Regarding the quality control, the most important technical parameter of anthocyanin extracts is the color intensity, which is measured in the EV units. The EV is the absorbance of as 525 nanometers of a 1% solution of the product in a pH 3 buffer. It is very important to keep this pH well standardized because some variations can lead to have an error in the determination. And other parameters, important parameters, are the hue index, which is related with the oxidation level, and the blue index, which indicates the blue shade respecting the red coloration. In this sense, according to this last parameter in Segna group, we are able to obtain products with a red shade or a blue shade. In the quality control, other parameters such as pH, density, total acidity, or humidity are also analyzed depending on whether the product is in liquid or powder form. Regarding the stability, the anthocyanin extracts is fundamentally affected by light, temperature, and the pH. However, anthocyanins from black carrot have a higher relative stability compared to anthocyanins from other sources due to the acylation of anthocyanin structure. Regarding the shelf life of the extracts, depends on the format in which they are manufactured. Here in Segna group, you will find two formats. In liquid form, which offers a different shelf life from maximum 6 months up to 2 years, depending on the aseptic or non-aseptic packaging, or the storage conditions. Or the other, the powder form, which is stable up to 3 years with packaging properly closed. As already known, as Antonio Morata explained, the anthocyanins have a unique natural physical property where changes in their pH will affect the color shade. The recommended pH range for the application of our anthocyanin extracts range between 3 and 4.5. As you can see in this slide, depending on the type of the anthocyanin that we use, we have a different response in terms of color and shade, and this is related on the pH of the product we are intended to color. For example, in the case of anthocyanins from grapes, by low levels of pH, the tone is red, changing to blue-purple by increasing the pH. In the case of black carrot, it is more stable to changes in the pH, maintaining the red color at any pH value, as you can see in the picture. However, the red cabbage anthocyanins have a violet color from the low pH levels, increasing in intensity when the pH grows. This is essential when you are looking for the color you need in your matrix, of which it is very important to know the pH. At Segna Group, we also have the necessary equipment and methodology to obtain the anthocyanin profile of the products that we produce. We perform this by using liquid chromatography, and in this sense, we can identify and analyze its purity and quality. On this slide, you have an example of a profile of a grape, black carrot, or red cabbage anthocyanin extract, and you can see how the distribution of the peaks is totally different and is defined for each type of product. In addition to extracts, we produce concentrated vegetable and fruit juices, which can be also used to color your food and beverages. These juices have a completely natural manufacturing process that consists of pressing the raw material followed by water evaporation. This is important to consider them as a clean label. So when using these products in your formulation, it is not necessary to include the E number as an additive. For concentrated juices at the Segna Group, we use different sources of raw materials such as grapes, black carrot, and red beets. Although we also manufacture other juices such as apple, pomegranate, and the strawberry, among others. The quality control of the concentrated juices is not related with the concentrated juices from grape. It's not related with EV, but is measured as units of color. This is defined as the absorbance at 520 nanometers of a 2% solution, of a buffer at pH 3.2. Another important parameter is the degrees Brix, which is the measurement of the mass ratio dissolved sugar to water in the liquid. In our laboratory, we measure it in a refractometer. For the black carrot juices concentrate, the quality control is the same that was explained for the extracts, but in color of red beet juice is measured in EV units. But unlike anthocyanins, here we use the absorbance at 535 nanometers, and the buffer must be adjusted to pH of 5. Regarding the stability, grape and black carrot juices are rich in anthocyanins, and the stability is similar to that explained for extracts. However, the red beet concentrated has betalains in his composition, and betalains have a lower stability to light, heat, or oxygen compared to anthocyanins. However, they maintain their coloring properties over a wider range of pH. This gives an excellent alternative to anthocyanins in products in which we want to maintain the red color at pH above 4.5, where the anthocyanins become blue and unstable. In the case of grape black carrot juices, the shelf life is the same than extracts. But when talking about red beet at a liquid form and refrigerated product, the shelf life is of 6 months, and for powder form is stable up to 3 years. Again, with the packaging properly closed. And finally, I'm going to give some examples of applications of our colorants in the food and beverage industry. First, we must indicate that the coloring extracts have a greater intensity of color and are purified. With a small amount, they provide a lot of color and don't usually affect with the aroma or flavor of the product we are coloring. And as we have said, they must be labeled as an additive with the corresponding E-number. However, concentrated juices have not such high amount of color as extracts. They usually require a greater amount to obtain the same effect, and sometimes they add flavor and aroma. The great advantage is that we can have a clean label. For example, both anthocyanins and concentrated juices from grape are excellent alternatives in wines to color or carry out fermentation process, or in liquors and alcoholic beverages to standardize the intensity of color in the production process. In ice creams and dairy desserts, the application is more limited when the pH is near or outside the recommendation. In this case, black carrot and anthocyanins or a concentrated bread juice are better indicated because they maintain the red color at higher pHs. In confectionery, pastry, and biscuits, they can be used, but care must be taken with the process in which heat is applied due to the natural condition of the colorants. And finally, for drinks, juices, and fruit preparations, both anthocyanins and betalains are an excellent option because the colorants are in their most natural condition. Well, that was all about the presentation. I just want to say thank you very much for your kind attention.
Well, thank you very much, Juan Mario, and everybody of the presenters, because we fit very well the time, and you are losing time. Next, we have 10 minutes to answer the questions to the attendees they sent us until now. We remind that to all the attendees that you can continue sending your question using the system chat. And if due to the limited time, any question remains without answer, we will answer by email in the few days. Then please, Antonio and Fernando, you can connect now. And we are going to make some of the questions that we have received. Perhaps it's not possible. We have received a lot of questions, but I try to read some of them. Well, the first is say good morning. I think it's addressed to Professor Antonio Moratas. Is how we can control anthocyanins reduction with the addition of SO2 in wine?
Thank you so much, Jose, and thank you so much, Michael, for the question. I have to say that it's not an anthocyanin reduction. The anthocyanins are there, however, they are as colorless forms. So these reactions is reversible and in the time is improving. However, we can control this reaction by using lower levels of sulfur dioxide, that in fact is other trend in the industry, but also using stable anthocyanin derived pigments, like for example pyranoanthocyanins that are not affected by sulfur dioxide bleaching. So it's a good possibility to use these derived pigments. Thank you.
Okay. Well, another question, I think in this case is to Antonio Morata too, is good morning. I have recently received demand for wine grape skins and seed extracts. It came from dietary supplement producer. Do any anthocyanins have any psychological performance to be used besides coloring? Thank you. I understand it's talking about health promoters or some additional benefits, not only coloring functionality.
Well, in fact, anthocyanins doesn't have so much nutritional properties. However, they have interesting antioxidative and healthy properties. So it is interesting to use to use them as natural pigments and very healthy molecules. So that is the other use. In fact, we can take anthocyanins at high amounts without any repercussions on health because we are doing it from a lot of years. And also, these molecules can be producing some repercussions on health because they have antioxidant properties. They can affect the vascular health, the blood pressure, they have anti-aging effects, and so on. But from a nutritional point of view, they have low repercussions as pigments. Thank you.
Okay. Well, we have another question. In this case is to Professor Fernando Gandia. Say, in your opinion, why are there so few food applications of betalains compared to other colorants? And what is the commercial future of betalains?
Okay, thank you very much. In my opinion, there is not that much information that we can find for anthocyanins. Betalains is a small group of pigments compared to anthocyanins because they are present only in plants of the order Caryophyllales. The scientific interest was behind because there was a small group of molecules. But now with all the biological activities that are being discovered for the betalains, I really think that there will be a bright future for the application of betalains in foods or in other sources, in other fields. The question is that we are only now learning things about betalains because we are only now able to produce individual pigments that we can use for performing the bioactive assays. And this is probably the main handicap that we are trying to reach now. This is the reason, the lack of information at a scientific level, but once all this is being studied now, they will promote the application in foods, for sure.
Okay, thank you, Fernando. And we have time only for two additional questions. This question is for Juan Mario Sanz. Is from a practical point of view, what are the differences between grape anthocyanins and carrot anthocyanins? When is not one question, is there are several. When should we choose one or other? Or what format and EV value are most suitable for the coloration of juice drinks?
Okay, thank you for your questions. Well, I think that the biggest fit differences between grape and black carrot anthocyanins are probably shade and stability. In the case of grape anthocyanins, have more bluish tone, especially at high pH levels. However, those from black carrot maintain the red color in these pHs. On the other hand, the black carrot anthocyanins have greater stability to change in pH and light, and also in temperature, due to their structure, as have seen in the presentation. In the second question, the best choice for grape anthocyanins is usually in wine-based products, and in those of black carrot, for example, when you need greater stability. And the last question for uses, the format and DV depends a lot on the characteristics you are looking for in the final product. The liquid format, it's the usually the most chosen, although the powder form is also available and is soluble in aqueous solution, and of course have a longer safe life. And as for the EV, it depends. It depends on the color that you want to obtain in the final product. For example, in our from our products, our most common standards are EV3 and EV6 for grape anthocyanins, and EV4 for black anthocyanins.
Thank you, Juan. Thank you, Juan Mario. And now is the last, we have no more time. This is the last question. It's about red beet, but I think it's more, it's not a question for Fernando, perhaps it's more a question for you. Is about the possibility of the commercial supply, but I'm not sure we have a good answer, but if not, I would like you to discuss. If not, we can answer now, we can send the mail after. Is a good morning. Can we have beetroot red powder with high betalain content more than 0.7%, or cannot be supplied? Thank you.
Well, this is, I think that's in the specification of our products. We have the betalain content, and this is something that sometimes we have to analyze in the lab. We can take note of the question and answer properly and better after the presentation if you want.
Well, thank you for your answer. I'm very sorry, but it's 11:12 unless here in Spain. But unfortunately, there is no time for more questions. And I say you at the very beginning of this presentation, if you have any additional questions, you can send them to our email address. It's not necessary if you send before by chat, because we are going to read and to prepare all the answers. And we will gladly answer them in the next few days. I remind you that you will receive in the same way the webinar documentation by email within 10 days. And I think that you can have a link of the recorded presentation if you want to review or send to some of your colleagues. And I hope you like the presentation, and see you again in future events. Thank you very much to all of you, and goodbye until the next time. Bye.
Bye.
Bye. Thank you so much.